The Baskin Lab is an interdisciplinary team of chemists and biologists at Cornell University. We are located in Ithaca, New York, which is a beautiful part of upstate New York that's surrounded by the Finger Lakes region. Our lab is uniquely a part of the Department of Chemistry and Chemical Biology, as well as the Weil Institute for Cell and Molecular Biology. We are situated in Weil Hall, which is an interdisciplinary life science and engineering building. The location of our lab at the center of Cornell's campus has fostered many cross-disciplinary collaborations that have been key to the success of our science.
Our research interests are at the interface of chemistry and biology, and the molecules that fascinate us are called lipids. These are the fatty substances that our cells and tissues use to insulate us from the cold, to store energy after a big meal, and to erect barriers called membranes that separate their insides from their outsides. Within these membranes, certain specialized lipids act as signals, kind of like turn signals in your car, that tell the cell when it's time to change course or to adapt to changes in its environment. There's a lot that we still don't know about how lipids act as signals within our membranes, and a better understanding of these phenomena could lead to new treatments for diseases where lipid signaling is defective, like Alzheimer's disease, multiple sclerosis, bacterial and viral infections, and many types of cancer.
We tackle these scientific problems from many different perspectives. We draw upon experimental approaches from building individual molecules to studying entire model organisms. We use the power of organic synthesis to build fluorescent probes for tagging lipids. We also engineer and evolve enzymes to manipulate lipid signaling. We have an in-house, state-of-the-art fluorescence microscope that we utilize to image the locations of lipids in cells and track their movement, to analyze the precise molecular composition of the lipids and identify proteins that they bind to. We use techniques such as mass spectrometry and HPLC to further complement our work in cells and to establish the physiological relevance of our findings. We use many model organisms, including fruit flies, zebrafish, and mice. Collectively, these tools have enabled us to identify important new roles for lipids in development and in disease contexts such as cancer.
Each member of the laboratory has the opportunity to present their scientific findings at conferences and symposia at Cornell, as well as national and international conferences. We have a lot of fun inside the laboratory and outside of the laboratory too. We have picnics, we go apple picking, we have bowling and virtual game nights, and we cheer on the Cornell Big Red hockey team. We also are dedicated to sharing our passion for science with others in the community through outreach activities that aim to inspire the next generation of leaders in science and technology. Thank you for watching, and for more information, check out our website or send us an email. After watching our general lab video, we want to introduce more details about one of the instruments in the lab that enables us to make new scientific findings, our state-of-the-art fluorescence microscope. A microscope is a laboratory instrument used to examine objects that are too small to be seen by the naked eye. A substance has fluorescence when it emits light after absorbing light that is higher energy. Many ocean animals that glow in the dark, like jellyfish, absorb energy from light in the environment through a process called excitation and emit fluorescence. They can do so because they make fluorescent molecules biologically. Fortunately for us, scientists have been able to isolate and prepare these fluorescent molecules in the laboratory to completely revolutionize the way we can study and visualize cellular structures with a microscope. In our lab, we use these fluorescent molecules to label membranes of cells and image the fluorescence with our microscope to study the biological properties of membranes. We will now show you some major components of our microscope. First, we see the eyepiece. This is what we look into to examine the samples we are about to image. The black knob on the side is used to adjust the focus, which means to adjust the distance between the objective lens and the sample. It is important to obtain a good focus so that our images are clear and not blurry. The black platform that we see now is used to hold our samples in a dish or on a slide. Underneath the platform are the objective lenses of our microscope, which are used to magnify samples. The microscope is connected to a computer, and we can use the computer software to tell the microscope to start imaging and display acquired data on the screen. We are going to do a small experiment that demonstrates how we use our fluorescence microscope to image membranes and DNA in cells. In these two dishes of cells, we have already attached green fluorescent molecules to the plasma membrane. We rinse off the media that cells grew in with a buffer to keep the cells alive and treat the two dishes with two different blue dyes, Hookstain and DAPI. For both dyes, if the molecule can pass through the plasma membrane, they will bind to DNA within the cell. In the next part of the video, the screen will be split into two halves. The left side will show the dish treated with DAPI, and the right side will show the dish treated with Hookstain. We put the dish on the sample holding platform and bring the objective lens up to the dish to image the cells. We observe the cells to the eyepiece and adjust the focus using the adjustment knob on the side. We select the proper excitation light for the samples and move the sample holding platform around to look for representative cells. We then monitor the fluorescence on the computer screen in live view. Under the green channel, we can see green fluorescent signal from both dishes. The signal comes from the labeled plasma membrane of the cells. We choose an area of interest and zoom in more, and finally adjust the focus more finely with the computer software until we reach a focal plane that we want to image. We then switch to the blue channel to look at the DNA dyes. We can only see a blue fluorescent signal from the dish treated with Hookstain and not from the dish treated with DAPI. At last we acquire images in both fluorescent channels, first blue and then green. Here are the images that we acquired through our microscope. On the left are cells from the dish treated with DAPI, and on the right are cells in the dish treated with Hookstain. Both images show the plasma membrane of the cell in a green color, illustrating the defined boundaries set by the plasma membrane. However, only DNA in the cells on the right image are stained blue because Hookstain molecules can enter through the plasma membrane, but the DAPI molecules cannot. These images demonstrate the semi-permeability of the plasma membrane. As we discussed in our demo video, besides the plasma membrane, cells have many other membranes that form different organelles to carry out a variety of vital functions. In the last part of this video, we are going to show images of the cell that are stained with 4 different fluorescent molecules to mark 4 different organelles in a group of cells. The structures shown in cyan are the nuclei of the cells that contain inheritable genetic material of the organism. The web-like structures in magenta are a part of the organelle called the endoplasmic reticulum, and this is the place where proteins that dock on membranes are produced. The small dots and circular vesicles shown in yellow are endosomes, which are responsible for transporting and passing along the proteins produced in the ER to other parts of the cell, like the plasma membrane that is shown in green. These membrane compartments collaborate closely to ensure that cells are functioning properly. The ER is like a factory that produces goods which are membrane proteins inside of the cell according to the blueprints provided by the DNA in the nucleus. After the goods are produced, they are loaded into the endosomes, which are like post office delivery trucks. The protein products are delivered to various destinations, including the plasma membrane, to carry out their distinct functions that allow us to do things like see, listen, smell, taste, feel, move, and think. Thank you for watching the demo and lab tour for Beyond Bacon: Why Fats Are So Important. We hope that we were able to illuminate some of the fundamental properties of membranes and demonstrate what life is like as a research scientist. We are so excited to be a part of one of your first adventures as a scientist.